Hello, my name is Kunjumon Vadakkan. I study the nervous system to understand its operations. I have a conflict of interest to declare. In the previous introduction video, I have explained the necessity for developing a new hypothesis and test that hypothesis. So, in order to introduce that hypothesis, first we need information about the normal functions that we already know. So, today I am going to explain a single neuron and its parts, and also the connections between the neurons called synapses. So, there are different types of neurons. Basically, they are classified into excitatory neurons, which excites the neurons in the next order. Inhibitory neurons, they inhibit neuron in the next order. So, let us see an excitatory neuron and its parts. So, an excitatory neuron will have a cell body, that is a cell body, and it has inputs coming towards it, and so this is a dendritic main stem, dendritic stem, and it branches out to form a tree called dendritic tree. It has a tree shape of a tree, and at the terminals of these dendritic branches we see structures called spines. So, this is a dendritic spine, dendritic spine. It's also called a spine, just symbol spine. So, that is the input. So, this is the dendritic spine, and similarly we see dendritic spines there on that dendritic branch. So those are dendritic spines. In other words, they are the input structures. The spine has a head region and it has a neck region. So the neck region has a lot of electrical resistance in that region. So now the potentials that are generated at these dendritic spines, they propagate through this dendritic branch and they arrive at the cell body. As they travel through this dendritic branch and dendritic stem, their strength get weakened. In other words, the potentials get attenuated. As they arrive at the cell body, they cross this cell body area and arrive at this axon. This is an axon, that is the output structure, and it goes, branches out, and it terminates into the output regions, which are called presynaptic terminals. So these are presynaptic terminals. So why they are presynaptic terminals? Because they synapse with the spines of the next order of neurons. So they synapse with the spine of this next order of neuron. So that forms the junction between the presynaptic terminal and the spine of the next order of neuron. So the spine is also called postsynaptic terminal. Postsynaptic terminal. So dendritic spine is otherwise called spine. It's also called postsynaptic terminal. In contrast to the presynaptic terminal, which is the output terminal of this neuron. And here comes the synapse, which is the junction between the presynaptic terminal and the postsynaptic terminal or the spine or the dendritic spine of the next order of neuron. Now let us see how this cell function. So this is a neuron, and the neuron generate spikes at different points. Spikes. What is a spike? Spike is a very high potential. It is generated by adding up small potentials. So for example, at the cell body, at this region of the axon, which is called axon hillock, it's called axon hillock, the potentials that are arriving from the dendritic tree to the axon hillock, the potentials summate or add up to generate a somatic spike. It is also called action potential. It's also called action potential. Once action potential is generated, that action potential propagates through the axon to all its terminals. Now, spike is also generated at the level of the dendritic spine area, which are called dendritic spikes. The strength of the somatic spike and the dendritic spikes are almost same, but we still don't know how dendritic spikes are getting generated. Dendritic spikes were discovered 15 years back. We don't know how a large spike is generated from the potentials, single potentials that are generated at these individual dendritic spines. Now, let us see what is going on at this junction, which is called the synapse. If you draw a synapse, the action potential, it propagates towards this presynaptic terminal, and there is entry of calcium into the presynaptic terminal, and within this presynaptic terminal there are synaptic vesicles with neurotransmitter molecules within them. The synaptic vesicles release its neurotransmitter molecules into the synaptic cleft. And the neurotransmitter molecules get bound to this postsynaptic membrane and elicits postsynaptic potential. It elicits postsynaptic potential. So in the case of the excitatory neuron, it's called EPSP, that is excitatory postsynaptic potential. In the case of the inhibitory neuron, it's called inhibitory postsynaptic potential, or IPSP. Now let us see during rest state. So here we have an action potential arriving here. During rest state, when there is no action potential, there are certain changes taking place at the synaptic terminal. From these vesicles, single vesicles release its contents into the synaptic cleft, and that is called quandal release. Quandal release. It's happening all the time. A single vesicle is getting its contents released into the synaptic cleft all the time. So that's called quandal release. That's happening even while rest, even while sleeping. So that's the default state of the system. When an action potential arrives, there is a volley of neurotransmitter release into the cleft, leading to postsynaptic potential, which is a high potential, which travels or propagates through this dendritic tree and reach the axon hillock. By the time it reaches here, it gets weak or get attenuated. So how many dendritic spines are present in an excitatory neuron? In the case of the monkey visual cortex, there are 5,600 nearly spines per neuron. So that is monkey visual cortex. Whereas in the case of the monkey motor cortex, there are 60,000 spines per neuron in the case of the monkey motor cortex. Now the question is, do we need to activate all these spines to elicit, to summate or add up the potentials to generate a somatic spike or an action potential? The answer is no. Nearly 40 action potentials, if they can arrive simultaneously at this axon hillock, it can generate an action potential. But in practice, as these potentials arrive at this cell body, they get weak. So it is possible that nearly 100 action potentials may have to reach from around 100 spines, and they summate spatially. That means summate at the same time at this space, at this location, to generate an action potential. Anywhere between 40 and 100 will be sufficient. So once action potential is generated, it propagates towards all the presynaptic terminals. Now the propagation is unidirectional, and it's also important to note that at the synapses there is a synaptic delay. Of 1 to 2 milliseconds. So that's a big delay compared to electricity, which travels at the speed of light. So here at the synapses, there is a synaptic delay of 1 to 2 milliseconds. Now, since nearly 40 to 100, around 40 or in between 40 and 100 inputs are required to fire an action potential. When we say a neuron is firing, we mean an action potential is generated at the cell body region. Now the question is, when we see that, what does it mean? It means nearly 40 or maybe up to 100 of its inputs elicited postsynaptic potentials that arrived at that cell body, summated together to generate an action potential. So if you look very carefully, there are very large number of combinations of these inputs that can generate the same action potential. Let's take the example of a neuron that has around 30,000 spines, that's somewhere between 5,600 and 60,000. Let us say it has 30,000 inputs. So if you ask the question, how many such combinations of roughly 100 inputs are possible from these 30,000 inputs? So how many combinations of 100 inputs that can generate an action potential are present within these 30,000 spines? The answer is, it's around 4.68 into 10 to the power of 289. So that many combinations of inputs containing 100 spines can generate an action potential from this set of 30,000 spines. So when we see a neuron firing, it is not at all specific with respect to its inputs. It's highly non-specific when we look at this gigantic number of combinations that can give rise to a single action potential. One thing I forgot to mention is that the condyle release of the vesicles from the presynaptic terminal induce a very small potential at some sub-segments on the spine head, and those potentials are not capable to traverse through the spine neck. So they lead to a depolarization of the spine head region, maybe a subcellular portion of the spine head. Now, an action potential will induce a postsynaptic potential which traverse or propagate through the dendritic branch to the dendritic stem and reach the axon hillock. And even though they get attenuated, if they don't get attenuated, if they are coming from very close to the cell body, then 40 such postsynaptic potentials are enough to summate to produce a somatic spike. But if they are coming from remote regions on the dendritic tree, then they get attenuated, and we may need nearly 100 of those potentials to summate to generate a somatic spike. So let us think that 40 postsynaptic potentials are sufficient enough to summate an action potential, because they are coming from close to the cell body. And if we draw a graph of the threshold, this is the potential, and if this is a threshold, 39 postsynaptic potentials won't get summated to generate an action potentials. So they will not generate an action potential. But when 40 postsynaptic potentials, that is nearly 40 postsynaptic potentials, summate spatially, that will generate an action potential. So this is an all or none phenomenon. All or none phenomenon. So in this context, even if 5,000 potentials arrive from 5,000 spines and summate at this axon hillock, they will generate the same spike. So in that regard, we can see that around 4,960 potentials do not contribute to the generation of an action potential. So this is a supra-threshold stimulation, whereas sub-threshold stimulation by 39 EPSPs also will not generate an action potential. So if you look very closely, we see that this does not look like an efficient system. We have to look at like that because we are still struggling to understand how the brain works, and we are trying to look for a mechanism that generates inner sensations. So the question is, do these 4,960 potentials generated at different dendritic spines, are they contributing to some other function that we don't know yet? Similarly, dendritic spikes, these are spikes that are comparable to somatic spikes. We don't know how they are getting generated. Are they contributing to the formation of inner sensations? So when I explain the hypothesis that I've put forward, I have found the contributions of these potentials as well as the dendritic spikes towards the generation of inner sensations. I think you have understood the basic operational mechanisms of the neuron and the synapses. If you have any questions, please feel free to ask me by sending an email or below this video. Thank you very much for your attention and have a great day.